So now I want to show you a more precise movie of how RNA polymerase performs transcription. This is an actual crystal structure of RNA polymerase, and you can see that the active site and the bridge helix, which is where the reaction of assembling of the RNA molecule from individual RNA nucleotides occurs, aka RNA polymerization. The reaction that RNA polymerase facilitates happens. And then we also have the typical polymerase shape with a clamp and a cleft through which the DNA template will be fed through. So of course, before RNA polymerase can go, you have to have various transcription factors bind RNA polymerase and thus activate the polymerization, the active site of the polymerase. So there's TF2, which is the transcription factor 2 needed for final transcription activation. It bound. Now we have the Tata binding protein and the closed DNA bind to TF2. You can see there's an association occurring here, and TF2B core has bound to the actual RNA polymerase. And then we have various things that shove the template into the clamp. Let's speed up through that. And here we have the first RNA polymerization event occurring. The DNA has been opened, and now we are synthesizing or polymerizing RNA right at the active site where the little pink magnesium ion is. So the movie will zoom in to the region right now. There we go. So you can see here that the magnesium is orienting the incoming nucleotide because the phosphates of the incoming nucleotides are negatively charged and magnesium is positively charged. And these guys are pushing the synthesized molecule, the RNA chain and the next nucleotide closer together so that a polymerization reaction can occur. There we go, there comes the new guy being held here by a pink magnesium ion. The magnesium is shoved over by the red loop, and now we can catalyze a formation of a phosphodiester bond. And the two ATPs here, held by the magnesium ion, are removed. And the reaction will repeat itself until we have enough RNA, until we encounter a termination signal. And here we have the alpha-amanitin, which is the toxin that's found in the poisonous mushrooms that blocks the reaction. And here, what is occurring here, what is being shown here for you, is the proofreading ability of the RNA polymerase. Again, I don't expect you to understand this at molecular level. I just want to show the actual process as opposed to depict RNA polymerase as a blob. Here you can see the RNA polymerase backing up when it made a mistake, cleave off something that it doesn't like, and restart again. Despite its proofreading ability, it still has a plenty high error rate for the compared to DNA polymerases, which are very, very careful. All right. Hopefully this was not too overwhelming. The link for this movie by Chang and Kramer is found under the Science Direct link. It's the actual article, but if you scroll down, you can get to the supplementary information for the movie, if you would like to take a closer look at it. And again, there is no sound with this movie, there is no narration. You'd have to read the actual article in case you would like to learn more. Okay, so now that we have made the RNA molecule and terminated the RNA transcription, we need to now prepare RNA for translation. In eukaryotes, it means that you have to do three important things. You have to splice, you have to add a 5' cap to the front of the molecule and a 3' poly A tail to the back. The only molecules that can actually exit the nucleus and be translated into proteins are mRNAs that have a 5' cap and a 3' tail. And if you'd like to have an extra point on the RNA quiz, give me a good reason why it is a good idea to process mRNA. Why is it useful for the cell to be able to process mRNA? Okay, so this black and white image actually shows an arrested RNA transcription process. In this case, the green guy, the green string in the middle is the DNA template, and now there's multiple RNA polymerases sitting on that DNA, and each one of them is making an RNA molecule. And what I would like you to see, so one of these red strings coming off the green template is the many mRNAs that are being produced. And what hopefully you can see here, that a lot of them have these loops on them. And that these are the sites of splicing, because and splicing is done by these really large complexes called spliceosomes. In fact, you can frankly see the huge U1, U2 complex sitting right there and popping out an intron, splicing the axons of an RNA molecule together. You can see one right here as well, the big blobs. And splicing, depending on the speed of the RNA transcription, splicing is usually done almost completely by the time RNA transcription is finished. So most molecules that come off the DNA template are fully spliced by the time the RNA transcription is done. And here's a closer look at the whole process. So as soon as RNA transcription begins at the start site, and as pol 2s are moving along the template towards the end, the first thing that happens is the 5' cap is added right here. Then splicing is occurring, and then finally, as the fully processed mRNA is done, all of the introns pretty much have been cut out, and it's ready for the next step of adding the poly A tail. So now let's look at intron removal via splicing movie that is also conveniently found on YouTube, but I have a downloaded version. Here, this movie is with narration, so you're welcome to watch it with narration as well.
It needs to be edited to remove.
So here we have the 5' end of an RNA molecule, and here in green is an intron, and in yellow are the axons. The spliceosome complex is assembled because both the 3' and the 5' end of the intron will be recognized by the smaller guys, and then the big spliceosome assembles and forms this lariat structure. Almost done here. There we go. The lariat has always been formed, is it? Almost, not quite. There we go. Now we've formed the lariat, and then it's very important for this, in order for spliceosome to function properly, not to mess up which end goes where. That is why the lariat of the intron is formed first, and then we ligate together the two axon ends to make for a perfect cut. So here we have 3 introns, and you will see how they each moved out from the mRNA molecule. And again, for the sake of simplicity here, the molecule is shown completely separated from the template, as if splicing occurs afterwards, but usually it occurs concurrently with RNA replication. Again, you can watch this movie with narration at this YouTube link. Now, one of the benefits of splicing is the fact that pretty much all of our human genes are alternatively spliced. What it means is that our cells can choose to incorporate or fail to incorporate different axons, creating alternative versions from the same gene, alternative versions of proteins. So in this case, there's 3 splice variants of a particular gene that is encoded in DNA. And once the RNA has been transcribed, and depending on specific splicing choices, you can either end up with all 5 axons, 1, 2, 3, 4, 5, or axon 3 is dropped in the shorter version of protein B, or axon 4 is dropped in this particular version of protein C. And this will produce, even though the first domain and the last domains of the protein will be the same, these 2, say, catalytic loops will either be present or absent, or maybe those are regulatory loops that this protein uses. And in certain cases, a regulatory loop can be missing, so a particular protein is then silent or unresponsive to particular types of regulation.